Yeah, so, like everybody said, thanks for coming in the bad weather. So, I'm going to tell you something a little bit different from what you've heard. It's more basic science. It's really about memory. And I'm going to be focusing on one brain structure in particular. I noticed it appeared in some of the slides, but I'm going to tell you a bit more about it. It's called the hippocampus. It's this little structure in the deep in the brain, in the medial temporal lobes. And it's commonly thought of as a memory area, and I'm going to explain that a little bit. So our modern understanding of this area started with the patient HM, who suffered, so as a young man, he suffered from debilitating seizures, epileptic seizures that he would have, I think, daily, it got to. And so he underwent surgery for this, and his seizures would begin in the hippocampus, so that the surgery was to remove the hippocampus on both sides. This was in 1953. So at that time, that was the surgery. And the side effect, though, was a complete anterograde amnesia. So HM, he could remember things from a long time before, his childhood, from his younger years, but he couldn't remember much from around the time of his surgery, and he couldn't form new memories. So the classic sort of scenario for HM was, he would have a conversation with his doctor, and he could act fairly normally with the doctor, talk to the doctor for 10 minutes or so. The doctor would leave, comes back 10 minutes later, and it's like he's never met him before. So we'd say, "Hello, doctor, nice to meet you. I'm Henry." If anybody remembers the movie Memento, it's actually a not completely unreasonable depiction of somebody with hippocampal damage. Subsequent studies found out that it wasn't just all memories that were affected, it was a subset of memories. So there were certain things that HM could learn, could remember, and they were mainly motor skills. So one of the experiments is to get him to learn to write his name backwards using a mirror, which is actually a little tricky if you try and do it. And so the classic result there is he could learn to do that. So the doctor would put the mirror in front of him and say, "Henry, can you write your name backwards?" And he'd do it perfectly. And then he'd say, "I've never done this before." He'd actually done it hundreds of times. So procedural learning is a slow process of getting better through practice. The kind of memory then that was associated with the hippocampus was called declarative, which is a difficult term, so I'm not going to actually talk about that because that was refined a little bit into this a newer conception, which is episodic memory. Now, episodic memory is now thought to be really what the hippocampus is for. And this is actually a scientist, Endel Tulving, a psychologist, who, in this funny-looking journal cover, who coined the term and described it first. But to really explain this, I'm going to need a volunteer. Who wants to volunteer? Well, I think you were first. You're all the way over there. All right, you've got to stand up. All right. I'm going to ask you a question, okay? You can stand. I'm going to ask you a simple question. What did you have for breakfast this morning?
I didn't have breakfast. Oh.
All right, that's good. That's good. We'll do that. What did you have for?
I had a banana and walnuts, actually.
Okay, good. Fantastic. All right. Did you know I was going to ask you that question? Have we ever met before?
I don't think so.
No. Did you, when you had your walnut, what was it, walnuts and banana, did you make a special effort at that time to try and remember and save that information and write it down somewhere and have it written? No. So that was something you automatically did, just like millions of other memories that you're forming all the time, even when you're doing particularly inconsequential things like just having breakfast. But more than that, I'm going to ask you a question, but I'm going to answer it, which is, how did you do it? And I think the way you did it was, you couldn't just tell me what you had, right? In fact, it took you a little bit of time. I think you had to go back in your mind and picture yourself in whatever it was, your kitchen or wherever you had the breakfast, and tell me if I'm wrong. You had to picture yourself, and you had to recreate that memory. You had to travel back in time in your mind, have that memory again, recreate it, and look and see, right, what's in front of me, what am I eating? Okay, Thank you very much. What's your name?
I'm Danny.
All right, Danny?
Yeah.
Thanks, Danny. So, this is what Angel Tulving said about episodic memory. I'll read it quickly. Remembering past events is a universally familiar experience and uniquely human. As far as we know, members of no other species possess quite the same ability to experience now, in a different situation, perhaps different form, happenings from the past, and to know that the experience refers to an event that occurred somewhere else. Other members of the animal kingdom can learn, benefit from experience, acquire the ability to adjust and adapt, solve problems, and so on, but they cannot travel back into the past in their own minds. So I think there he makes a very vivid description of what this kind of memory is, but I think he's wrong. Actually, I'm going to be showing you some data from rats, which I hope will convince you that we can study this in animals. And of course, as we've already learned, there's great efficacy from being able to study mental processes in animals, both for understanding just how the brain works and understanding how it can go wrong in disease. So there's another interesting notion that's recently come out which does relate, and it's called a default network. So one of the things that many neuroscientists do is they image the brain in living people in things like MRI scanners. And you can do this functional MRI where you can get someone to sit in a scanner and ask them to do various things and see which bits of the brain light up. What that actually means is they're looking at a blood oxygenation signal in the brain, which kind of correlates with which parts of the brain are most active. So you can sort of say which areas of the brain are particularly involved at any given time. But this accidental discovery was made by Marcus Raichle, that in between the tasks the experimenters were setting, where the person wasn't really doing anything, just daydreaming, it wasn't as if the brain was just defaulted to some random state, but there were particular areas that were particularly active, and active in a way that varied together. So there's really a network of areas, and the hippocampus is really part of this. So it's the hippocampus and various areas that are strongly connected to the hippocampus that form this default network. So there's a growing combination of views that the hippocampus and associated structures might be involved in memory, but also in, shall we say, daydreaming in general, thinking, putting yourself in other situations here, free thinking, remembering the past, envisioning the future, and even considering the thoughts and perspectives of others. And of course, there's good reason to study the hippocampus because it can be affected in many diseases. You probably know that Alzheimer's is associated with memory loss, amongst other things. And you certainly have damage to the hippocampus and connected structures. Hippocampus is also very vulnerable to, for example, strokes where you have loss of oxygen. It's a site for medial temporal lobe epilepsy. It can be subject to infection. Schizophrenia is associated with hippocampal damage as well, in some cases, and depression, extreme stress, and aging. So there are lots of, the poor hippocampus is under attack from lots of different sources. So let's take a closer look, and let's take a closer look in a rat, because then you're going to see that I'm going to be able to show you some more details. We can really look inside the brain with the rat and find out what the hippocampus is doing. And firstly, the hippocampus in a rat looks very, very similar to the hippocampus in a human. It's a very, very conserved structure. This is a relatively early structure to have evolved in mammalian evolution. It's a much bigger proportion of the rat brain, but it has that characteristic look. It's like two bananas in the brain. And this is looking from the top, from the side, and the back. See the structure. It's been known about for a long, it's been studied for a long time. This is actually a venerable diagram of what kind of a cartoon of what you get when you slice, if you slice the hippocampus, you can see the structure of the cells inside it. This is drawn by Ramon y Cajal about 100 years ago. And what you can see is this beautiful architecture, at least you might be able to see it so easily, but there's a particular loop of information where information comes into the hippocampus and proceeds through connections between different groups of cells in different areas. And there are many exciting things about this structure, but one of the things is It's a beautiful structure to record activity from. And that's what I'm going to be talking about in this lecture, actually. Recording the activity of brain cells in rats that are actually running around and making memories and using those memories. And so what we do is we drop our electrodes into this area so that they're close to cells in this area. It's called the CA1 area of the hippocampus. And we listen in on how these cells are communicating to each other. So, neurons, these excitatory neurons, they fire spikes, which are electrical impulses. They can fire many tens of impulses per second, but actually their activity can be much more finely timed than that. But these impulses are how they communicate to all the other neurons and in these dense networks. We listen into these impulses and try and understand them. They're like a code. They're like the information moving around the brain. We try and understand what is being encoded, and can we see memory in these spikes from these cells. So that's really what we're trying to do. So we drop an electrode into this group of cells. The electrode just nestles close to the cells, and it pushes them out of the way and hangs out in the extracellular space. The first problem we have immediately is you're surrounded by hundreds of cells that are all firing action potentials, these electrical impulses, all at the same time. So how can you make any sense of it? The trick we use is to use a special electrode called a tetrode, which is really just 4 wires twisted together. Now, only the tip of the wire is essentially insulated, so only the tip is exposed. And then these 4 wires are twisted together, and now we're doing a special trick. These wires can each pick up signals from all of the cells around, but because one cell will be closer to one wire than the other wires, we can distinguish cells that are over here from a cell that, let's say, is over here, which have a different pattern of activity. The strength of the signal we pick up here is related to how far the cell is away. So we can see all the impulses, but we can also say something about where near the electrode those impulses are coming from. And just to demonstrate that, this is a little cartoon of what we see on the electrode, the small spike. But if we just pick off the height of this spike, which is related to how close, the strength of that signal is how close the cell is. As the cell is closer to the electrode, you get a bigger spike. Now, if we just take 2 of these wires, and every time we get a spike, we plot it as a point in this 2-dimensional space. So bear with me. You've got 2 values, the height of the spike on one channel and the height of the spike on the other channel. That 2 numbers is like an x and y coordinate. We plot all of those coordinates. You see that there are clusters. And the reason there are clusters is because there are different physical sources of the signal. So we're using this procedure to triangulate the signal and separate out the cells. Why do we do it? Because what the cells are doing is very interesting. So this is one of the first things that was discovered about cells in this area. You're looking top down on a say 2 meter by 2 meter arena. You're looking at the path of a rat just running higgledy-piggledy around this arena over the course of about 20 minutes. And that's black. On top of that, in the red, I've put a little dot wherever the cell, one of these cells, and there are millions in the hippocampus, one of these cells has fired a spike. And you can see, you should be able to see, there's a very strong spatial representation. You only get a spike whenever the animal passes through this point. And he can come through it from many different directions at many different times throughout this recording session. This cell only cares about this place. We call that a place cell, and we call this its place field. The point is that all the different cells that you record from have all different favorite locations. And so across a population, you've got what's been called a map of the environment in the head of the rat and monkey and human. So this has been called a cognitive map, but I want to show you some of the properties. For a long time, this was hard to understand. It's a very interesting phenomenon, but it's hard to understand, I would say, as a model of memory, because you have to go back to the place to get this cell to fire. Let's say you form a memory here. Every time the rat's here, let's say we do something, we give him a bit of food. All right, so now you could associate this cell with food. The problem is, if the animal wants to remember it, he's got to activate that cell somewhere else. Otherwise, he's got to go back and get the food, but maybe he's trying to find out where the food is. This doesn't help if he has to go back there to retrieve the information. So I just want to illustrate that with an imaginary maze. Imagine you are a rat in a maze like this. Right? You'd have to find your way. What do you want to know? All right? I'm going to put words in your mouth here and say, you might get out, and then you want to remember, what did I just do? Because you might end up, the experiment might put you back in about 5 seconds' time. So you want to remember, how did you get out? Or maybe you're stuck in the middle and you want to look ahead you want to consult the map in your brain and look ahead and find out, what do I do next? The problem with place cells, as we understood them, is they only tell you about where you are. They're a map where you only get to look at the current location. And so the whole issue is, how could these cells be informing about memory when they're really stuck to the current location? Now, we did know for a very long time that the hippocampus actually operates, in a rat, operates in 2 different brain states. There's a very characteristic rhythm, 8 hertz, which means 8 times a second. And this rhythm is something you can record from just a wire dropped into the hippocampus, where you're actually recording from all of the cells. It's a very global signal. It's like brain. You've heard of brain waves. I mean, this is not now about single cells. This is about just all of the cells averaged together. But they're governed by the strong 8 times a second rhythm whenever the animal's running around. And that's when you see the place fields that we talked about. But the animal just has to stop for a few seconds and immediately goes into this completely different mode of activity, which is characterized by long periods of relative silence, punctuated by periods where it looks like the whole hippocampus is active together. And to cut to the chase, this is our default network activity. This is the model, we think, that the rats have it just like we do. That the hippocampus, when it's engaged, it's in this activity, but when it's disengaged, and the animal is capable of doing memory or daydreaming, he's in a different state, and now I'm going to tell you what happens in these states. And when you look more closely at these sharp waves, they have these interesting rhythms. I'm not going to say that. So we developed a task, and now I'm going to skip over a huge amount of research that's established that place cells will be active at times other than when the animal's in the place field. For example, in sleep. There's a lot of research about activity in sleep. I'm going to skip over all of that in the interest of time and tell you about a recent experiment that we did. We created a memory task for these rats. So now they're in this 2-meter big environment, and they're looking for chocolate. They love chocolate. And we have chocolate at 36 possible little locations in this arena. They have to find their way to the chocolate. Of course, we want them to do it lots of times. We want to get them to explore around a lot. So we play this game on them. We have the chocolate appear in a predictable location, and the rat runs there. And then we put the chocolate, these little wells, so we can control the chocolate coming up. Then we put the chocolate somewhere else, and he doesn't know where it is, and he's got to search all around to get it. As soon as he finds it, he's allowed to go back here again. Then he has to look again everywhere, and then he can go back here again. So we have these 2 modes he's toggling between: goal-directed, memory-directed navigation, and random foraging. And you can see that this is just saying it takes him a lot longer to find the random wells, so he can't smell them. We know that he can't find out where they are. He doesn't know where they are. See what the next slide is. Right. So then we had to record a lot of cells. And I'm really going to sort of not spend too much time on this, but we developed a technology which basically made all of our electrodes and everything smaller and lighter, so we could put a lot more electrodes on the animal's head than had previously been put. And the result of this was that we could record these little place fields that I told you from hundreds of cells simultaneously. So this is showing you the firing fields of individual hippocampal place cells, but where we've actually got 212 of these cells. Some of the cells fire everywhere, There are actually known to be physically different kinds of cells called inhibitory interneurons. We can exclude them from the analysis. But all the other cells are classic place cells and fire only in particular regions of the environment. And all these things are simultaneously available, right? So they're all doing this. But this is a lot of data to analyze, so what are we going to do with it? Oh, this just shows you, it blows that up a little bit, so you can see these individual place fields. The rat is only about this big on this scale. So these are pretty big representations, but they all overlap, and I'll show you what that means. So in order to understand what the hippocampus is doing, we have to decode this activity. We've got a lot of activity from a lot of cells. We've got to try and understand it. How do we do that? We developed this scheme where we basically pretend to be some brain area listening to the hippocampus. And we say, well, what would that brain area do with this information? So we say, all right, let's say there are 4 cells that have place fields that look like this. And let's say that they each just fire 1 or 2 spikes. What are we going to infer from that occurrence? And we say, well, let's look at where the place fields overlap. If you get a couple of spikes from each of these cells, we're going to have a fairly strong belief that the animal is here, right? Does everybody sort of understand that idea? Because if you just listen to one cell, you really wouldn't know where the animal was because the cell has a pretty big firing field. But by looking at multiple cells, you can really narrow it down to a small area. But the killer, feature of this is you don't have to decode the current location. If it so happened that a bunch of cells fired and collectively represented some other location, we could have a strong belief that this location is being represented, even though the animal's actually over here. And then it doesn't always work, so if we got random activity, we wouldn't have any strong overlap, we wouldn't have any strong belief. So now I'm going to show you, hopefully this will work, some movies. This is a real-time movie where you can see a rat, his position and direction as he's running around in this arena and exploring. And you can see the 36 food wells, and in red you can see the home well, and green is actually a well that has the food currently. I'm going to show this again. What you can see flickering behind is the decoded representation of space by the hippocampus, and it tightly follows where the animal is. There's actually a color associated with every single pixel across the screen, but only those that are close to the animal are strongly represented. So the hippocampus is really representing the animal's current position at all times. But what we're interested in is what happens when the animal stops. Right? He runs a little bit, he stops. He runs a little bit, he stops. And this is what happens when he stops. This is what happened immediately afterwards. So now you see that the representation of position in the brain moves smoothly away from the current position and off towards the remembered goal position. And these are 4 sharp waves that occurred immediately. You like that? 4 sharp waves that immediately occurred. And during each sharp wave, there was this trajectory. But there's one thing I didn't tell you. This is speeded up. So these events, the first movie was real time. These are speeded up by a factor of 20. So each of these little imaginary sequences is occurring 20 times as fast as reality. So you can actually, I don't know if you can see the time going by. It's just a few hundred milliseconds are going by for each event, if you see that time value. So we can chop this up into frames, and at each frame, the representation of position is very tightly focused on a particular position, but squashing it across time, there are lots of these events that depict trajectories. And the animal's stationary throughout these events, and they're basically happening all the time whenever the animal stops. I'll just show you a few things about this. We take all the events that happen when the animal's at the home well, and we actually just superimpose all of those figures that you saw in the previous slide, and they have one location particularly in common, and that's the current location, because you tend to get a bias. The sequences tend to start from where the animal is. But what's cool is if you take all the sequences that start from the other locations, the random wells where the animal's thinking about coming back, they also overlap in the goal location, showing us that these sequences really do reflect a memory for the goal. And another way of looking at that is just to say what's the total probability that's shown for each of the locations, and it's actually a statistical outlier. So this means that as an experimenter, I can just look at this activity and I can tell you where the animal's remembered goal is, without looking at its behavior. So we can decode that memory. And when we move the goal to a different location and he slowly learns it the next day, we can also do that trick. There are a couple more things. We determined that, yes, the activity tends to match the future direction, the future trajectory the animal's going to take more than the past. So there's the sequences can occur in lots of different sequences, but they really match what the animal's about to do. But finally, I want to explain one more thing, which I think is an interesting feature. We designed the task, actually, so that we moved the goal position every day. So the animal had to relearn what was the remembered location. And we cycled through a bunch of random locations that don't repeat during the day. You put those two things together, and what that means is every trial is a novel trial, in the sense that the animal's not had to start in that particular location and go to that other location. The combination of start and end point is novel on at least the first 19 trials of every day. So we have this interesting data set where the animal's continuously being forced to generate novel trajectories. And this just shows an example of a movie. Now we combine behavior and these sequences, so we're flicking between timescales, but you'll get the idea. So the animal finds the random well, and then this is a trajectory he's never done before, and it's depicted perfectly. Then, interestingly, you get another trajectory. This is quite common. Remember where that was. Then you get another one that is the intended movement. Then the animal does it. He gets his food. And then you remember what he was thinking about before. Well, he thinks about it again, and then he goes there. All right, and this just says, yes, the novel sequences were just as well predicted as the others. So, summary, hippocampal neurons can fire in sequences that match the past trajectories, but also sequences that predict future trajectories and remembered goals. And these predictive sequences can even imagine trajectories the animal's never taken before. All right, so now, why do you need sequences? All right, so it looks like a model of memory, but can we say anything about whether, put any more evidence towards that this really is important for memory? And we ended up doing a study that addressed this issue essentially kind of backwards, but we ended up looking at an interesting mouse, and this is just a cartoon, imagining a mouse that has the ability to sort of look forward and back in time, and a mouse that doesn't, and why that might be a bad thing. We ended up looking at a mouse that was a mouse model of schizophrenia. Now, there are many mouse models of schizophrenia, and it's also the case that you can never convince somebody that you really have a mouse with schizophrenia, and that's reasonable, right? What would it even mean? But there are some interesting behavioral features that are associated with schizophrenia, including things you might not think of. So, for example, there's certain very basic features like startle responses. If you make a loud noise, we all jump. But if you precede that loud noise with another noise, which is not as loud, you don't jump as much. But people with schizophrenia do. They can't turn it off, and they can't learn that stimuli are irrelevant. And so those kinds of things were replicated in this particular mouse model. But the other feature of this mouse model is that I'm going to really just summarize this in words. The connections between cells were plastic, are plastic in general, but in this case, they were plastic in a skewed direction. There's a tendency for them to get stronger and not weaker. And this idea of potentiating and depressing the connections between neurons, they're both important in the normal brain, and in this brain, in the hippocampus, and in areas of cortex, there was an imbalance towards too much excitation. So it's an interesting mouse to look at. But the mouse had severe behavioral problems, and one of them was working memory. So in a task very similar to the kind of task I just talked about, where the animal has to remember to get back to a goal, this mouse wouldn't be able to do it. Kind of interestingly, I wouldn't say amazingly, but interestingly, that rest activity that I've spent all this time talking about was really different in these mice. They had too many events. So those sharp waves that I talked about, there was an excess of events, which you might expect because there's too much excitability, right? So whenever the animal pauses, we measured this by looking at a particular frequency band of what we call the EEG, that global activity, but it's too excited at this level. But interestingly, when the animal was actually running around, there wasn't this overexcitability. So we think these are the replay events, the trajectory events I talked about. This animal has too many of them. But then when we looked at the place fields in this animal, they were completely normal. So while the animal's running up and down, the size of these response fields that you would expect, the amount that you get, the firing you get within them from each cell, the tendency to fire more in one direction than another, and various other measures were completely normal. As soon as the animal stops, so he runs, everything's normal. He just stops, and everything goes haywire. Because I already said he has too many of these events, but within each event, each cell is firing too many spikes. And when you combine that together, too many events, each with too many spikes, it multiplies. It means that in general, there's an almost six-fold level of increase of excitability, but only when the animal pauses. So it's specific for this default activity or memory activity. Oops. And finally, we wanted to say, well, what about replay? Unfortunately, or what I've called trajectory sequences. Unfortunately, it's very hard to get that data without recording from all the cells that I showed you, and mice are smaller. So the electrodes you can put in them and have them carry around is far fewer. But we were able to get some handle on it by looking at pairs of cells and saying, well, if two cells represent fields that are far apart on a track, they should fire far apart during these sequence events. And if they represent fields that are close together, they should fire close together. And although it's kind of noisy, in the normal animals, you get this relationship, this sort of V curve here, where the place fields are close together, the firing in these events is very tightly locked, but it gets looser as the place fields are further apart. And there's this relationship you get. In these animals, it's completely gone. In this mouse model of schizophrenia, they have no relationship between where the place fields are, basically, and how they fire during these events. So they're overactive during the rest events, and the rest events have lost all of their spatial information. So I'll summarize. The place cell sequences may have a function in working memory, in this kind of memory ability to put yourself in a different situation and access information from somewhere else so that you can figure out what to do. And they may provide a model system for looking at the effects of brain diseases such as schizophrenia on memory and cognition. So with that, I will thank, first and foremost, the members of my lab, including Brad Pfeiffer, who actually is being coy here, is at the back, but he did 99% of the movies and the trajectory story that I told you about, collaborators and funding. Thank you.